had 2 cups of coffee. Sorry.
Hey guys. Okay, first of all, this is, before I read the other announcements, this is the IR spectrum that I posted on Moodle for polystyrene. So it's the standard IR spectrum for polystyrene. Don't print it out. We don't need everybody to print out the same spectrum. Just use it for analyzing your IR spectrum, as well as you've got a peak list there to include in your table, the IR table for the analysis of your polystyrene IR spectrum. But again, don't print that guy out, okay? So use the peak list, use the information there. And it's on Moodle. It's also, there's a copy in each of the organic labs and near the IR room too. So while you're collecting your IR spectrum, you need to reference they're posted around, all right? Let me go through some announcements really fast, and then we'll get started with talking about the Grignard experiment. So this week, experiment 8, the Grignard experiment. Make sure to read that experiment very carefully. Lots of instructions in there that you've got to follow very carefully. So make sure you read it and reread it a few times. You'll have a quiz this week, and then your experiment 6 report is due this week. Next week, on the syllabus, it says we're doing experiment 10 and 11. The actual doing is experiment 11, okay? Experiment 10 is going to give you information about gas chromatography, but the actual doing is in experiment 11, and you're going to use your alcohol from experiment 8 to then go on and do another reaction with it in experiment 11. So remember what your alcohol is for your pre-lab, because it's specific to that half sheet of paper you got last week for writing the pre-lab for 11. And then next week your experiment 7 report is due, which is another big report like experiment 3. Everything goes into your notebook. So make sure you have your notebook all ready to go at the start of lab period next week. Make sure you get, for experiment 7, make sure you get GPC data from your instructor. So we went to try and run samples for GPC samples for the Tuesday lab people, and we couldn't get the instrument to work properly. So we never ran any student samples, so we have example data that you're going to analyze for your experiment 7 report. Make sure you get that data from your lab prof. Now I want to talk a few minutes about registration, because this week's advising week, and next week you're going to be registering for classes for the spring, and talk to you about Chem 256 and what is Chem 256A, what is Chem 256B, and things you need to be aware of with both of them, okay? So if you need to take organic lab next semester, everybody needs to register for Chem 256A. If you are taking lab next semester, you need a Chem 256A lab, okay? That's the required portion of second semester organic lab. It'll be a lot like this first semester thing. All your instructions are going to come out of the lab manual. The experiments will get a little more complex. The data analysis will get more complex. They'll be basically kind of the same thing as what you've already been doing this semester. Chem 256B is not required, but you have to take Chem 256A before you take Chem 256B, is the independent project portion. And so you will write a proposal for a 3-step synthesis, turn in that proposal before spring break, and after spring break you'll carry out that 3-step synthesis until the end of the semester, okay? And so you don't have to do that portion, but in a lot of cases it's a good idea to do that portion, and it's a good way to get a research-like experience if you are interested at all in research, or interested in seeing if you are interested in research or not, because it is a very real research-like experience, okay? For Chem 256A and B, if you're going to take a Chem 256B, you need to register for the 256B that corresponds to your 256A. So you want the same day, same time, same professor that you had for Chem 256A. The one thing to be careful of is Chem 256 section 5A is taught by Dr. Valdez. That is only for the first half of the semester portion. There is not a 256B that goes with it. So if you are registering for that lab section, make sure that you don't want to do the independent project, because the only people that should register for that lab section are people who are definitely not doing the independent project. If you at all think you want to do the independent project, you should register for one of the other lab sections, and there is another corresponding lab section on the same day as Dr. Valdez, okay? So section 256, section 5A is only for people taking the lab for 1 credit, and no, they're definitely taking it for 1 credit for the A portion, okay? If you aren't sure about 256B yet, but are at all thinking about possibly doing it, register for it now, because we need to know the numbers now of who's interested in it, versus later on we may not have a spot available, okay? So make sure that you register now. If you are at all interested in 256B, register for 256A, and the same professor day time for 256B, okay? Any questions you guys have? All right, if you guys have questions as registration goes on, contact me, you can contact your lab prof, we can all help you out. The one other thing to keep in mind is if you end up waitlisted for a section that you need to email me with, because I take care of all of registration and all of waitlists. You need to email me with what section you need, and it's got to be a need, not a want, when we're dealing with waitlists, okay? So what lab section you absolutely need because of your class schedule, and then why you need that lab section, okay? And then make sure you end up on the wait list for that lab section, because I can't do anything with wait lists to help you get into that lab section if you aren't on the wait list. Then you have to wait till drop add to do anything, okay? So if you need a section that fills up before you can register for it, email me with your need and why that is a need, or if you have like 2 sections that you could get into, that's always helpful to know too, because then I have a little more flexibility getting people into lab sections. But email me and get on the wait list for the section that you need. All right? Okay, so let's talk about the Grignard experiment. Lots of things to cover with that guy today. So it is called the Grignard reaction because someone discovered it. So it is, when you write it, make sure you give it a capital G, not a lowercase g. Victor Grignard in 1900, so this reaction's been around for a long time and still is an important reaction in organic chemistry, discovered this reaction, and so it is named after him. The first part of the reaction is not just the Grignard reaction. We actually have to make what is called the Grignard reagent, and you guys are going to do that this week, okay? And so what is involved with making that Grignard reagent is we are setting ourselves up to be able to use the Grignard reaction, which is really important in organic chemistry because it involves carbon-carbon bond formation, which doesn't happen that often in organic chemistry. So when you find a good reaction that you can get 2 carbon-carbon bonds that weren't there before, it's an important reaction. So to set this up, we're going to start with an alkyl halide. We're going to use an alkyl bromide. Bromides tend to work really well. Iodides, okay. Chlorides, okay. Usually people are using the alkyl bromide. We're going to use ether as our solvent. We'll talk about here in a minute why that is. But what happens is you make this reagent that involves actually kind of an insertion here of the magnesium into our alkyl bromide. And so this is, this guy right here is known as an organo magnesium halide, and in this case bromide. And what we get with this is we actually end up with something that has a partial positive charge on the magnesium because it's a metal, and so to counteract that, we're going to have a partial negative charge on the carbon, which is also unusual. Usually, if anything, carbon's going to have a partial positive charge. So that will help us with the reaction. And so you end up with MgX, and again, in our case, it's going to be a bromide. So we've got partial positive on the magnesium, partial negative on our carbon. And so we're going to use that to be able to use that carbon as actually a nucleophile in the reaction. So like I said, this is really powerful. The reagent then reacting in the reaction is really powerful for organic synthesis and putting carbon-carbon bonds together. That carbon it's not a really strong nucleophile, so you get the carbon is a nucleophile, but it reacts with weak bases, okay? So it's not like it has a full negative charge there. And so some of the things that it can react with are water, and so we'll talk about that because that's going to be important. Something important we want to avoid. It will react with alcohols. It'll react with aldehydes, which is what we're going to use. It'll react with amines. These type of things, it'll go. Sometimes you can get the Grignards to react with ketones instead of the aldehyde. So it does actually, that carbon does act like a nucleophile though when we get to the reaction itself. The reason that we use ether in this reaction and in the reagent formation is it helps to solubilize the magnesium, okay? It also has a high vapor pressure, as we know from experiment 3. And so that helps keep the oxygen out. And that's going to be important for this reaction. So here's an example of this reaction. If we had bromobutane, and we react it with magnesium, which some of you will, then you're going to get this guy here. Here. So you'd get this magnesium bromide, so the butyl magnesium bromide out of it, okay? So as far as setting up what's going to happen with your Grignard reagent, this is pretty easy setup for figuring out what's going to happen, okay? And since we're involving something that's an organometallic reaction, we've got organic and a metal. At this point, we're not going to show you the arrow pushing that's really involved either. It's more understanding where the partial charge is for the reaction, or for the reagent formation. Okay, so just with this guy, some of the side reactions we need to be careful of involve things we've already kind of talked about. And so I'm going to discuss the side reactions and tell you what we're doing to prevent these from happening. So one thing that we've talked about is this, our Grignard reagent can react with water, which we don't really want to happen. So when it reacts with water, what you're going to do is kill your Grignard reagent, and then basically you form the alkane corresponding to your alkyl bromide, plus then this guy. Okay, so if you, in this case with our bromobutane, we'd form butane, and then this bromomagnesium hydroxyl species afterwards, okay? So what we're going to do is we're going to use dry ether and glassware to prevent this from happening. Then next thing they can react is we've talked a little bit about keeping oxygen out. So this will look similar to what we want in the Grignard reaction, but we don't want it at this point. And so we're preventing oxygen with the vapor pressure of the ether. We're also going to use cotton in the openings of the glassware to prevent the oxygen from coming in, okay? And there's a couple other things we'll do as well. We'll talk about it in here in a little bit, but we're not even going to stir our reaction right away to kind of help prevent water and oxygen from getting in. Then another thing that can form is you can actually have your two alkyl groups reacting together. So we could have two butane groups reacting and form octane, and this forms from adding your alkyl bromide too quickly. To the magnesium. And so if you get this guy, and he then is going to react with just alkyl bromide. So we formed alkyl magnesium bromide, and then we've still got alkyl bromide around. We'll form magnesium bromide out of that, okay? And so here, we're going to keep the water out to prevent this side reaction. Here, we're going to keep the oxygen out as best we can to prevent this side reaction. Here, we're going to add our alkyl bromide dropwise to our magnesium so we don't ever have a high concentration of alkyl bromide that's not reacting, okay? All right, so let's talk about, we'll look at the equipment setup, and we'll talk about some, because this guy has a unique setup that you haven't used yet. We'll talk about some of the precautions with this setup, okay? All right, so we're going to have, this apparatus is what you're going to use for your Grignard reaction, okay? And you're going to have your stir plate, and then you're going to set this up with some space in between your round bottom flask, okay? And I'll tell you about why that is in a little bit here, okay? You're going to use your 3-neck round bottom flask, clamp on the neck, and then you're going to use your Liebig condenser and clamp loosely on that condenser, okay? Then you're going to have a stopper, a glass stopper here with just a little bit of grease in here so it doesn't get seized with the ether. You're going to have your SEP funnel with, make sure the stopcock doesn't end up in the oven again. Then you're going to put cotton in the top of your SEP funnel when you're done, and you're going to put cotton in the top of your condenser when you're done. And your condenser you use just like you did with reflux, water in, water out. Okay, you're going to connect the water. So you're going to start with, first of all this glassware is going to go in the oven before lab this week. So the morning labs at 7:15, we're going to start loading the ovens, and we want them loaded before lab starts at 7:30. In the afternoon labs, you're going to want to come in before your afternoon lab and get it all loaded before 1 o'clock, okay? So make sure you're on time, because we can't start counting the heating time until the oven is closed permanently and we're not adding anything more to it, okay? And if you're late enough, we won't put your stuff in the oven. Once you've gotten everything out of the oven and it's cooled down, you're going to set up this apparatus, okay? You're going to have your stir bar in your round bottom flask when it goes in the oven, so it should be there still when you are setting this apparatus up. And then you're going to, once you have this all set up, then you're going to add the magnesium to it, okay? And you have to calculate how much magnesium you need, okay? It doesn't tell you in there how much. You have to figure that out. Then you're going to put 10 mils of ether over that magnesium, right? Then what you need to make is a 0.1 molar. You're going to need 0.1 moles of your alkyl bromide. Okay? And that's going to be in 40 mils of ether. So you need to figure out with your specific alkyl bromide for your reaction, how much in volume is that 0.1 mils, okay? And so what you're going to do, an easy way to make up that solution without having to then get like an Erlenmeyer flask out and stir it and have it introduced to water and air and so on and so forth, is put your ether in your sep funnel first, okay? And make sure your stopcock is closed, all right? Then if you pour your alkyl bromide, which is more dense than the ether, it's going to pass through the ether into the bottom of the sep funnel, and that'll get it mixed fairly decently, okay? And if you needed to, you could kind of jostle your sep funnel a little bit just to mix it around. But you don't want to get a lot of oxygen in there either, okay? So if you just put your ether in first, and then put your alkyl bromide in here, make sure your stopcock is closed, okay? So at this point, before you do any adding of this solution to this solution, or this solution to this mixture, you're going to have your apparatus checked, okay? So when you have magnesium, 10 mils of ether in your round bottom, you have your 40 mils of ether and your alkyl bromide in your sep funnel, but don't add anything yet, you're going to have your apparatus checked, okay? Then what we're going to do is check your apparatus. We're going to throw in some iodine. So the iodine helps to, magnesium reacts with oxygen pretty easily, makes magnesium oxide, okay? We need to clean the surface of the magnesium so it'll react. We need to get rid of that magnesium oxide, and that's what the iodine does. It'll clean the surface of the magnesium. So we're going to throw a crystal or two of iodine in here, and then we're also going to put a small portion of this mixture in your reaction, okay? And then what we need to do is get the reaction going, all right? So you need to start seeing signs of it reacting. It'll start turning cloudy down here. The color of the iodine will dissipate. You'll start seeing bubbling on the magnesium. All of those are signs of reaction. And to help spur that on, you're going to actually warm this apparatus with your hands, okay? Because ether doesn't have that high a boiling point, and it doesn't need that much heat to get the reaction going, okay? So you're going to warm this guy with your hands, watch for signs of the reaction, and then you're going to start adding this solution dropwise, okay? And you, throughout the addition, you want to add it dropwise. And what you're going to be doing by adding it dropwise is controlling what's going on in the reaction. So we're adding it dropwise so we don't get our alkyl portions to add together, but we also need to control this reaction because it's an exothermic reaction and we don't want it going all at once, okay? So we're going to add things dropwise, and that's our sep funnel in this case is actually going to be used as what's called an addition funnel instead of a separatory funnel. So you keep adding it dropwise. You want to keep a nice reflux going in your mixture, but you don't want it going out of control, okay? So if you have questions about is your reflux getting too excited, talk to your lab prof, have them take a peek at it before you add more alkyl bromide to it, okay? But what we don't want to do, we don't want it too excited, but we don't want to get it to the point that it wants to die out, because once it dies out, it's really hard to get it going again, okay? So we want to keep it going, but we don't want it going out of control. The other thing you're going to want next to this setup is have a pan of cold water with maybe some ice in it, just in case it gets a little bit warm, okay? But again, don't use that pan of cold water until you've consulted with your instructor, okay? Because we don't want your reaction to stop. Because again, once it stops, it's really hard to get it going, and it's really hard to continue on with the next step, because this is just the Grignard reagent formation, then we need to go on to the Grignard reaction, all right? The other thing with this setup, we are not stirring. So we have not started stirring. And the reason for that is we don't want to bring in water and oxygen from the atmosphere into the reaction, okay? So no stirring yet, all right? Okay, so now we're going to get into our Grignard synthesis. So we're going to use our reagent and actually then do a reaction with that reagent. And so you're going to be reacting your Grignard reagent with an aldehyde, the aldehyde that you have on your half sheet. So we've got our Grignard reagent here already in that round bottom flask, okay? And one thing too, once you've added all your Grignard, all of the alkyl bromide solution, you're then going to heat your reaction for 15 minutes. You want it refluxing for 15 minutes, okay? So either keep it refluxing or use hot water bath to keep it refluxing, okay? Either way, keep it refluxing. So either with or without heat, keep that going for 15 minutes, okay? Then, and still no stirring. Then we're going to work on this part of the reaction, okay? So we're going to add our aldehyde. And again, everything is still in ether, okay? So what we've got so far is we've got that partial negative charge on our carbon from up here, okay? Now that's going to actually attack. So this carbon, that partial negative charge, is going to attack our aldehyde, okay? And we'll get the movement of electrons. That nucleophile will attack that carbon, and then we'll get the movement of electrons up from our double bond of our carbonyl, okay? And so what we are forming, you want to keep track of your who's who, okay? So we've got R1, we've got R2, and we've got our oxygen is from our carbonyl of our aldehyde. And now we're going to form the species similar to what happens when you get a reaction with the oxygen, okay? But now we've got both parts on here, so now that is okay. And now we're going to have the partial negative charge on the oxygen and still a partial positive charge on the magnesium, and then we still have our hydrogen here, okay? So then we are going to work up this reaction with, we need acid to do that, and we're going to use sulfuric acid, okay? And so we get that flow of electrons as far as the reaction is concerned. Okay? And so then we get this alcohol that's formed. So we've got R1 is from our Grignard reagent, R2 is from our aldehyde. We form our alcohol that we want, okay? And this is where we want to stop for this feed. We don't want to go any farther than that, all right? What we don't want to do is we don't want this, okay? And so when you get to the point that you're adding the sulfuric acid, you have to do it very slowly and keep everything cold so that you don't get elimination product instead of just protonating our intermediate and getting the alcohol, right? So you can also eliminate with sulfuric acid instead of just forming the alcohol. So you could form this and then go on to form this. And we don't want this week, we want this next week. So for this reaction, we've just finished adding our alkyl bromide. with hot water bath, we were refluxing. Okay, now we're going to go to a cold water bath. And we don't want an ice bath, we just want a cold water bath. If it's an ice bath, it's too cold, you won't get reaction. Okay? So cold water bath, we are still not stirring yet. Okay? Then you're going to get 25 milliliters of the aldehyde solution for your particular aldehyde. Okay? And this is already made up in bottles in the hood. All right? So you don't make this solution. This is made up for you. The 25 milliliters of aldehyde solution is going to go into your separatory funnel again. We're going to use it as an addition funnel again. So make sure stopcock is closed. Okay? Once you have your aldehyde solution in the separatory funnel and you're about to start adding it, then you can start stirring. Okay, so we have not stirred at all yet until this point. You can start stirring and then adding the aldehyde dropwise. And again, we are going to control the addition. We don't want things out of control, going in too fast. We don't want things going heating up too much to create a reflux. So control that addition. Now, one thing you may notice with this is, and it's okay that this happens, is sometimes when the aldehyde is reacting with the Grignard reagent, it makes this popping sound. You'll hear popping in your round bottom flask, and that's okay. Okay, but you want to keep it a dropwise addition. Don't add it all at once. Keep adding it slowly and controlled, so you keep the temperature of the reaction controlled, and you don't get too much aldehyde in there all at the same time. All right. Okay, so now after that addition, then you're going to stir for 15 minutes at room temp. Okay. So you'll take the cold water bath off, stir for 15 minutes at room temp. Once you've done that part, now you're ready for the workup of the reaction. Okay? And this is going to involve us adding the sulfuric acid. So this flow diagram is in your lab manual. This is the flow diagram you want to use for your pre-lab. Okay? Because remember, the flow diagram is always showing the workup of the reaction, and this is the workup we're going to follow. Okay? So we've got our secondary alcohol, organic impurities, diethyl ether, and magnesium in there. And now we're going to start adding the sulfuric acid to quench the reaction and protonate our alcohol. Okay? For the workup, or we could call this the quench, we are now going to use an ice bath. And this comes from when we are adding the sulfuric acid, we do not want the reaction refluxing at all. Okay, so not even a controlled reflux. We don't want any reflux because we don't want it so hot that you're actually making the alkene after you form your alcohol. Okay, so you want an ice bath, and again, you're going to add dropwise addition of your H2SO4 mixture that you're going to make up. You're going to start with 9 molar sulfuric acid to make this mixture, so be careful with it because it's only half dilute. So concentrated sulfuric acid is 18 molar, you're going to use 9 molar, so it's still really concentrated, okay? So use gloves when you're handling it, be careful handling it, don't spill it. If we spill it, we need to clean it up right away so people don't get burned by it, okay? Then you're going to follow the instructions in your lab manual. With that 9 molar sulfuric, you're going to use water and ice to make the acid solution you're going to use for the quench. You're going to add that dropwise, okay? And like I said, you're controlling temperature here. Now, one thing you may see is it's going to, I'm going to go through here in a minute, the different reactions that are going on during this quench. One of the things it's doing is reacting it's basically killing or reacting any unused magnesium way back from the Grignard reagent. It's going to react with it. So you'll see magnesium spinning around and then bubbles coming off of that magnesium, and that's okay, all right? That's part of what's supposed to happen. But make sure you really control the heat with that addition. So let's look at all the things that are happening with that quench. So first of all, we are quenching our reaction. So we formed our intermediate from the Grignard reaction, and now we're going to add acid to it, and we are going to get the alcohol that we want, plus a magnesium bromide species that this salt's going to be floating around. It may end up floating, consumed in some way. But basically, you're quenching to form the product that you want. The other thing it's going to do is kind of kill some other stuff that's still potentially floating around. So if we have any Grignard reagent, it will take care of any remaining Grignard reagent. And what you will get is, remember this is from the Grignard reagent, you get the corresponding alkane to your alkyl bromide, and then again, some form of this inorganic species, okay? It's also, like I said, going to be reacting with any unreacted sulfuric acid, or sorry, unreacted magnesium. What you are going to form is magnesium sulfate, and then this is where you're going to get the bubbling. You're going to have hydrogen gas given off. That's going to be the bubbling that's going on the pieces of magnesium, okay? This can end up, this is a white solid, and so what you may start seeing is accumulation of the white solid. Don't worry about it till you're at the end of all your sulfuric acid addition, okay? Because sometimes what happens is you accumulate the white solid, and then eventually once you've added all the sulfuric acid, it goes back into solution again, okay? But you may see this white solid. When you need to worry about it is when you are done with the quench and all of your magnesium is done reacting such that you're now going to transfer to the separatory funnel. Then you want to make sure you don't have a lot of that solid still floating around. You want it all dissolved, because you're going to, when you go into the separatory funnel, it'll clog your separatory funnel, okay? So make sure that white solid is dissolved by the time you get to that point. And follow, there's instructions in your lab manual for how much water to add to help dissolve that, all right? Other things to watch for is just make sure the bubbling is done before you're going into the sep funnel. Make sure this reaction is done before. You probably won't see really these two happening, but you want to make sure this guy is done. If your magnesium is done bubbling, but there's still magnesium present, what you can do is then filter with gravity through the last wool. To get the last little bit of magnesium out before you go into your separatory funnel. Okay. Now, there's a couple things that we're going to talk about here that have to do with definitions of things for organic lab workups and things that you're going to continue to use. So keep these definitions in mind. You'll keep using these throughout the rest of the semester. So one of them is what's called extracting, okay? So we've got this mixture. We're putting it into our, this whole mixture, we're putting it into our separatory funnel. We're going to separate our organic layer and our aqueous layer, okay? Then what we're going to do is what's called extracting our aqueous layer, okay? So you're going to put your organic layer to the side, extract your aqueous layer with ether. And this time, so we've got the dry ether you use for setting up your reaction is in the reagent hood, so make sure you use that for the first part when we're setting up, when we were setting up our reaction. Now we're going to use red can ether. for the workup, because now we've added water, it's okay if things get wet. So with extracting, that means we're taking our aqueous layer and making sure we get rid of all of the organics that are still residually left in that aqueous layer. So that's the extracting, pulling the rest of the organics out of that aqueous layer. Okay? Then the next thing we're going to do, in extracting, that means we're going to have our aqueous layer, add ether, shake, vent in your separatory funnel, separate the two layers. All of that is implied in extract. Okay? Then we're going to take our organic layer and we're going to wash it with sodium bicarbonate. Okay? We don't want any of that residual acid around, because it'll still form alkene at this point, and we've got to do a distillation. So we're washing, we're taking our combined organic layer, so this organic layer and our previous organic layer, combine them together, we're washing or getting rid of, away from that organic layer, something we don't want, which is the acid, okay? Again, with washing, you're going to take your organic layer, put it in the sep funnel, put sodium bicarbonate in the sep funnel, shake and vent a few times, let the layers separate the two layers. So all of that is implied in wash, all right? Okay, at this point, make sure you've got either a neutral pH or a basic pH, which with bicarb, you're only going to get up to 8 with basic. But you want neutral or basic. You don't want it acidic. If it's acidic, wash it again. Make sure that you get to neutral or basic, all right? So then, what we're going to do is use what's called a drying agent. And we're going to dry our organic layer, okay? So we're going to remove residual water from the organic layer. And what we use to do that is magnesium sulfate, but we don't want that magnesium sulfate. We want anhydrous magnesium sulfate, okay? And so anhydrous magnesium sulfate is a white powder. It looks a lot like flour. And it reacts with water. And when it reacts with water, so we've got this white powder, and then when it reacts with water, forms clumps. So when it gets hydrated, it forms these clumps of white solid. So you're going to add magnesium sulfate until you've got, when you're swirling, you've got clumps that form with a little bit of free-floating white powder, okay? Now the key with this is you don't want to make pancake batter, so don't add so much magnesium sulfate that you can't even swirl it around. Then you've got so much magnesium sulfate in there, you're more than dry, and now we've got another problem of getting your organic layer away from it, okay? So you want to add enough magnesium sulfate that when you swirl it around, you've got clumps with some free floating. Then you're going to let it sit for 5 or 10 minutes, and then you're going to filter off the magnesium sulfate with gravity filtration. Rinse that solid with red can ether to get all of your product away from it. And then that magnesium sulfate and that filter paper is going to go in the waste container that's in the reagent hood. Okay? One thing you don't want to do with magnesium sulfate, and I know your lab manual tells you use about 2 grams or so of this. It's trying to just give you a ballpark number so you have an idea of how much you need. You're going to add it based on what I told you. Have enough in there that you've got a clumping with a little bit of it free floating, and when it's free floating, it's going to kind of look like a snow globe. So when you swirl it around, you'll have the free floating looking like a snow globe. You don't need to weigh this out, okay? You never need to weigh out magnesium sulfate. You're just adding it till you've got the appropriate amount in there, all right? And then, like I said, you're going to use gravity filtration to remove it. After after the gravity filtration, then we need to do a simple distillation to remove the ether. This is the point that you need to stop, or not need to stop, you need to get to this week, okay? You need to get through the simple distillation this week, all right? So this is our key step here, through that simple distillation. Then what we're going to do, and if you can get through this point, even better, we need to do a fractional distillation in our all-in-one simple distillation apparatus to purify our product. If you can get that done this week, great. If not, you want to do it over the week in open lab. So you'll have to, if you're doing it in open lab, you need to let someone know, make sure you're working out with your lab prof that it's a good time for you to be working in open lab. Or you potentially will have time at the start of next week's experiment to do this, okay? So if you can work efficiently in lab next week, you can get this potentially done next week before you start experiment 11. With this fractional distillation, make sure you know the atmospheric boiling point of your particular alcohol that you're making, okay? Make sure you know the atmospheric boiling point, not one under vacuum. So it's going to be in the hundreds, not something less than 100. So if you find a boiling point less than 100, you've got one that is under vacuum and you need to correct for it to be at atmospheric pressure, okay? So make sure you know that boiling point, and like I said, it's going to be in the hundreds or 100, above 100, not below 100, okay? So when you're looking up for your physical data table, make sure you get that correct number. And I think it was last year, Chem Spider was not giving correct information, so don't use that one to figure it out. Okay? Then next week we're going to do a GC sample. We'll talk about that. And the other thing that you need is an IR spectrum of your alcohol. So before you go on to experiment 11, you're going to need an IR spectrum of your pure alcohol. You will compare that. So for your literature spectrum that you're going to compare it to, you need to look up a spectrum of either your alcohol that you're making this week or something that's structurally similar, okay? So say you're making 4-octanol, but you can't find 4-octanol's IR spectrum. Well, 3-octanol's IR spectrum is going to be very similar to 4-octanol because they're very structurally similar. If you have 4-octanol and you use 1-octanol's IR spectrum, it's not going to be as similar because you're talking about a primary and a secondary alcohol. So the spectrum, especially in the fingerprint region, is going to look a little bit different. So if you can't find the IR for your product, use something that is structurally similar, okay? But make sure you collect that before you go on to experiment 11. So keep that in mind for next week. You need your IR spectrum, GC sample prepared. We'll prepare those in lab next week before you go on to experiment 11, okay? All right, any questions you guys have? Okay, so now if you want to stick around,
Where you guys going?
Dr. Gillmore gets potentially a little bit of time.
Go to Pine Prof last Friday.
Let's freaking
We
go.
missed out. Well, guess what? If you stick around for like 5-10 minutes, you can see Dr. Gillmore get pie.
Okay, so did you have it tied?
He had 100.
Oh, it's rough.
Okay.
Did he earn his tie?
Yeah.
Okay.
Okay, if you're leaving, be quiet so I can announce. That'd be great. All right, Dr. Gillmore. Try. So Dr. Gillmore had $100.60 of donations in his name.
Yes.
Really for life. Woo, yeah. And so that means Dr. Gillmore is $9.40 away from getting a second pie. So if any of you have a dollar you want to put in. So you give him another pie. You can put your name in and get drawn to pie him yourself.
I think whoever puts in the biggest donation right now gets to deliver the second pie.
I mean that too.
We draw for the first one. But
I do accept IOUs if I know you. So I can track you down. Nobody?
see, they love you.
Amber's got it.
Oh.
Yeah.
Friends. There goes that record.
We're friends.
You deserve this.
She would know. She works for me.
Wait, does that mean I get to buy him?
Do you want to do the first one?
Yes.
Okay. Alright, so, oh, I didn't bring the clip. Yeah. Okay.
I've heard some more shirts.
You've done this before, so remember if you're the one stipulation, the next person, the person who gets to do the other pie, if you pie him too hard, we can pie you back. So we're not breaking professors' noses.
Also, he's not allowed to say we pied him too hard. He wants to pie me.
No, if I get to pie anybody who wants to know. I had bet Janelle that she would not raise $100. That she was going to take a pie had you guys not gotten it.
Funny that happened.
David, you're taping this?
Oh yeah.
Why are you here? You're just not in Orgo.
I tape every week.
Video guy.
Oh, what? Okay, the video
Alright.
guy doesn't matter. Okay.
Let's get going.
Get it.
Alright. Regina Tan.
Oh.
Is she here? Oh,
That's me.
that stinks. Lauren Archer. Oh my word. I didn't even rig that. That just happened.
I know.
Wow.
That's right.
Alright.
It's a hard one. Oh, this is.
I do. Alright. The faculty got into this one here at third. Jeff said that. He's like, oh, I know.
You're enjoying the waitress?
How do you open this?
Let's go. Do the whole can. It's
That's
healthy.
why we're bad, right? You have to at least do it.
I know. That's my problem.
That's the point.
You ready?
Sure. That's
Organic
probably what you call
students
the
everywhere.
Lecture and lab, all the frustration.
I know. I
Just for
don't
all
know.
of us.
Oh well.
Thank you guys.
Thank you guys. Keep supporting Relay for Life.